Hi, thank you so much for coming today. This is my biology presentation going over chapters 16 and 17, covering DNA and RNA replication, transcription, and translation. My name is Lily Maves, and I am in period 1 of AP Biology. First off, what is semi-conservative replication? It describes the mechanism of DNA replication in all known cells. Semi meaning partial or incomplete, and conservative meaning keeping the same or to conserve. DNA replication, quote-unquote, unzips the DNA strand and creates an identical strand of the original. It actually conserves the DNA and its information to continuously be used and to be re-replicated. So in this model, you'll actually see that we have our original parental genome, and it's actually going to be replicated to create two identical ones, and it's actually going to continually conserve that original genome, and that's what it means by that semi-conservative. Now, origin of replication is actually a particular sequence in a genome at which replication is initiated or started. So basically where that replication begins on a DNA strand, and that's actually going to be right here on this diagram. Now, a replication fork is actually a structure that forms within the long helical DNA during DNA replication. It's actually created by helicases, which break the hydrogen bonds holding the two DNA strands together in the helix. And the resulting structure actually has two quote-unquote prongs, each made up of a single strand of DNA. Now, it's called a replication fork because you can see in the diagram, it actually splits out like a fork in the road or a fork that you eat with, in order to have those two prongs going off of your original strand of DNA. It's splitting in half. Now, our single-strand binding proteins, or SBB, work to hold and pin down each side of the replication fork so each strand of DNA can serve as a template for its duplicate. So binding proteins actually just help to bind and hold down the DNA strand so they don't move, so it's easier for it to be transcribed. Topoisomerases are actually enzymes that participate in the unwinding of DNA, so they help to unwind that helical structure that we originally have in our DNA, that double helix. It helps to unwind and straighten it. And they're used to DNA's natural helical structure, so it actually is required to be untwisted before replication occurs. Next up, we have a primer, and a primer is a short nucleic acid sequence that provides a starting point for DNA synthesis. And the actual primer, similar to like a paint primer, serves to prime and lay a foundation for DNA synthesis. So it just helps prepare and make sure that the DNA strand is well primed to go through the transcription process. And so you can see that we actually have our RNA primer right down here. And then primase functions by synthesizing short pieces of RNA that are complementary to the single strand of DNA, which serves as its template. And primers, as we stated earlier, actually must be synthesized by primase before the DNA replication can occur. So now we have the leading and lagging strand. Within each replication fork, one DNA strand, called the leading strand, is replicated continuously in the same direction as the moving fork, while the other lagging strand is replicated in the opposite direction in the form of Okazaki fragments. Now, Okazaki fragments are actually short sequences of DNA nucleotides which are synthesized discontinuously and are actually later linked together by the enzyme DNA ligase to create the lagging strand in DNA replication. So this is going to actually be our replication fork right here, but these are going to be our leading strand. So our leading strand is actually copied continuously, denoted by this continuous line, while the Okazaki fragments are in short bursts or discontinuous transcription processes on our lagging strand on the 3' versus the 5' end. Now, DNA polymerase 3, the main function of the third DNA polymerase is actually the duplication of the chromosomal DNA, while most other polymerases are involved in the repair of translesion of DNA synthesis. And it actually works with DNA helicase and a primase to participate in the replication apparatus that acts as part of the replication fork. And DNA polymerase 1, as stated earlier, is an enzyme that participates in the process of prokaryotic DNA replication. The role of the enzyme is actually mainly, as we stated earlier, to repair any damage with the DNA, but it actually also serves to reconnect those Okazaki fragments because they were discontinuously copied. They need to be put together again. They actually delete the RNA primers, and replacing the strand with actual DNA. Now, transcription and translation. Transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, or mRNA. The newly formed mRNA copy of the gene then serves as a blueprint for protein synthesis during the process of translation. Now, translation is actually the process of translating the sequence of a messenger RNA or mRNA molecules to a sequence of amino acids during protein synthesis. It's going to create a protein chain. The genetic code describes the relationship between the sequence of base pairs and a gene and the corresponding amino sequence that it encodes. So up here we have our transcription processes. Our transcription processes actually is the process by which going through the actual DNA strand and creating that RNA strand, that mRNA. And what that's going to do, our mRNA strand is actually going to go through what we call a ribosome to actually be translated into the polypeptide sequences or our amino acids. Now, messenger RNA, or mRNA, is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. The mRNA is an RNA version of a gene that leaves the cell nucleus and moves to the cytoplasm where proteins are made. And actually, during protein synthesis, an organelle called a ribosome moves along the mRNA, reads its base sequence, and uses a genetic code to translate each 3-base triplet, or codon, into its corresponding amino acid. Now, as I said earlier, we have our original transcription up here, and it's actually going to create our RNA transcript right here through our RNA polymerase. It's copying down and creating those series of 3 codons along what we find on our original DNA strand. And it's creating that messenger RNA that's actually going to leave our nucleus and go to places like our ribosomes, where we can find those. And ribosomes are actually a small organelle involved in the process of making proteins, which is called protein synthesis. The ribosomes handle translation, which is the second part of protein synthesis. And ribosomes can actually be found floating freely in the cytoplasm of a cell or attached to the place called rough endoplasmic reticulum, which actually is surrounding the nucleus, because this is our nucleus right here. This is our rough endoplasmic reticulum. Now, the anatomy of a ribosome actually consists of a large and small subunit, and what it's actually going to do is it's going to allow this little canal right through here for our mRNA to actually go through and be translated. So it's actually going to help read this strand of RNA, and it's going to create a protein based off of each set of 3 or a codon. Now, a codon is actually a sequence of 3 nucleotides which together form a unit of genetic code in DNA or an RNA molecule. And a codon actually corresponds with a specific amino acid or stop signal during protein synthesis. So you'll actually see that right along here, along our little RNA strand, you'll see that each 3 on here consists or corresponds to another amino acid or polypeptide sequence. So these are all of our polypeptides, and each one of these codes for one of their corresponding. Now, our reading frame is a way of dividing the sequence of nucleotides and a nucleic acid, either DNA or RNA molecules, into a set of consecutive triples. Each of these triples is a codon and corresponds to amino acids or stop signals during translation. So actually, our reading frame is actually going to be that set of the mRNA that our ribosomes are able to read at one time. And now RNA polymerase synthesizes RNA by following a strand of DNA. RNA polymerase is an enzyme that is responsible for copying a DNA sequence into an RNA sequence during the process of transcription. Now, as I said earlier a couple slides back, RNA polymerase actually is what makes the RNA. It follows that DNA strand and it transcribes everything that on those codons from our original DNA strand to create that RNA. Now, promoter sequences are DNA sequences that define where transcription of a gene by RNA polymerase begins. Promoter sequences define the direction of transcription and indicate which DNA strand will be transcribed. And initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. So the initiation is basically just the start of that transcription process. Elongation is the stage where the RNA strand gets longer with the addition of new nucleotides. During elongation, RNA polymerase walks along one strand of DNA, known as the template strand, in the 3' to 5' direction. And this is actually where those codons are going to be matched and create that strand of RNA that will leave the nucleus. And termination, conversely to initiation, is when it stops, when transcription stops. And so it stops translation and it actually terminates, it stops. And then we have our TATA box. A TATA box is a DNA sequence that indicates where a genetic sequence can be read and decoded. It's actually a type of promoter sequence which specifies to other molecules where transcription begins along that DNA strand. And last but not least, we have wobble. Wobble is actually an unorthodox pairing between the bases at a 5 prime end of an anticodon and the base that pairs with it. So it's actually pairing codons to their anticodons. So see here we have our nucleotides going from like adenine to uracil or guanine to cytosine, or where the wobble position is actually placing that G to become that cytosine. So that's going to be it. Thank you so much for watching my presentation. I hope this was helpful.